Welcome to the first episode of the Nervous Breakdown podcast, hosted by the Serious Brothers. Let's get into it, shall we? The first thing, which is very important to consult, is the difference between the white matter and grey matter areas within the spinal cord and the brain and their difference in function.
So the first thing to mention is, straight up, why they call it white matter and grey matter is just because of the way they present on imaging. White matter is the myelin, this wrapping of this proteoglycan, this lipid soluble membrane, many times around by your oligodendrocytes in the CNS and your Schwann cells in the PNS. So there, it presents as a white entity on your imaging, and your grey matter is then your cell bodies. So your cell bodies are your grey matter, your white matter is your axons, and your white matter tracts are your long projections of many axons bundled together through the brain, travelling together on these highways.
So the white matter acts as a connecting path for the grey matter areas.
Yeah, really. So you have your distribution, where you have your cell bodies around in the brain, and your white matter tracts take your information from your cell bodies, take it down in synapse into deeper structures, and then you have ascending tracts as well. Again, your axons are your white matter tracts, so they're literally what they take your inputs from your dendrites coming in from your cell bodies, and they pass it on down through your axons to your axon terminals, where you would have your synapses onto your dendrites.
Okay, so one of the key grey matter areas is the anterior grey horn, which has alpha motor neurons within it, which are very important in bridging the gap between neurons and muscles with the neuromuscular junction. Now, while we know what may happen after the anterior grey horn through the neuromuscular junction, something very important to note is how we get a nervous response from areas of the brain such as the primary motor cortex and premotor cortex, but mainly the primary motor cortex, down to the anterior grey horn and the alpha motor neurons and to the muscle cells. So could you explain, I think, is it the corticospinal tract that links these pathways?
Now, the way that the brain works is you have your primary motor cortex. This is where you initiate all your voluntary somatic movements. Your primary motor cortex also has associated primary association motor cortices and somatosensory motor association cortices, and these take in inputs from somatic sensation, from your position in space. All of these inputs from even other areas of the brain, such as the thalamus, such as the cerebellum, giving inputs on coordination, balance, position. All of these, they come together and they're brought through. All these are relayed into the thalamus, and then you have your descending tracts. So these all contribute of your upper motor neurons, going all the way down into your alpha motor neuron in your anterior grey horn, and then these will synapse out into your lower motor neurons, which will take your stimulus, take your responses down into the periphery. You have your primary motor cortex, you have your axons going down through your corticospinal tract, down into your pyramid, through your pyramids, into your alpha motor neurons in the anterior grey horn, and then synapsing out into your lower motor neurons.
Okay. Quickly, what is the official name for the nervous response that involves voluntary movement?
Voluntary movement?
Yeah. So could you briefly discuss the differences between somatic and is the involuntary? How would you call that?
So your somatic movement is the control from your central nervous system out into muscle that your brain controls. You are perceiving this movement. You can control your muscles contracting, you can control your movement in space, I can control the way that I'm talking. The other distinction you have is your autonomic nervous system. Now this takes care of your underlying homeostatic processes. It can also contract muscles and affect, but it also has effects on glands. These are effector organs. Muscles and glands are your effector organs. So these will be involved in your reflexes. Reflexes are very important in maintaining this negative feedback, either from, let's say, a sharp like a painful stimulus in your periphery where you have to move your muscles away quickly without you even thinking about it, really bypassing the brain, and also your internal homeostasis, so your breathing, your heart rate, your gut motility. All of these are controlled with your autonomic nervous system, which has two divisions. You have your sympathetic and your parasympathetic. So your sympathetic is your flight or flight, parasympathetic is your feed and breathe, more in particular. So it's really a contrast. There's more detail in this, but I don't know which way we're going to go down in this Synapse symposium.
Okay, I have some research here, written by myself.
Oh, interesting. And what is this article called that you've published? Or submitted to publishing?
It's called the EPQ.
Oh, quite. It's a big task to undertake. I did one myself. I wasn't looking so much at synapses though. I was looking at the genetics behind neurological disorders and how you could target them through gene therapies and protein therapies.
So this this action potential, we want to propagate it from the alpha motor neuron to the muscle cell.
To ask what happens in a synapse.
Yeah.
So your action potential is propagated down your neuron. Once you get to the bottom of the neuron and you've got this depolarization in your membrane, you open voltage gated calcium channels. Calcium then moves into your cell through these channels. A high concentration of calcium, what it does is it's sensed by synaptotagmins. We'll get into that. Calcium basically will cause vesicular fusion. So your upper motor neuron will then release acetylcholine, and then this acetylcholine will be released into your synaptic cleft, will then go and bind to nicotinic acetylcholine receptors on your postsynaptic neuron, your alpha motor neuron in this case. Sometimes muscarinic, more often than not nicotine acetylcholinergic. Bind, and then, which is an ionotropic channel, your NACHRs, ionotropic channel, get influx, calcium, sodium in particular, a little bit of potassium, influx alpha, it doesn't really matter. Basically it will cause your depolarization and you'll build up this graded potential, which will then be propagated into an action potential that will travel down your motor neuron.
Is there a specific junction between the upper motor neuron and the brain directly?
Yes.
What would that look like?
All your movements go through your thalamus. It's a relay station for sensation, your sensory stuff, and your motor movements. So they will pass. So you have a synapse in your thalamus from your upper motor neuron, and then your thalamus will relay out to your primary motor cortex.
Acetylcholine is the neurotransmitter that transmits impulses in chemical form across neuromuscular junctions, but it is not a protein. What is acetylcholine structurally? How do you make it?
Acetylcholine is an ester. It's an acetyl group. It might be easier if I get up a picture of acetylcholine. But basically, acetylcholine is here. Share this tab and stuff. Okay. So here you've got your ester group. You've got a small amine group here. 3. You've got tertiary amine there. So what is this metabolism, this build up and this breaking down of acetylcholine? Acetylcholine is formed by acetylcholine transferase, where you transfer an acetyl group from acetyl-CoA onto a choline molecule. You form your acetylcholine.
Choline is found in the diet.
Acetylcholine then, through VACHT channels, is pumped into your endosomes, so your vesicles. So these utilize a proton gradient, pumping hydrogen in actively, so then you can exchange out hydrogen for your acetylcholine, and you've got acetylcholine in your vesicles. Then acetylcholine released, and then you have on your postsynaptic membrane in the brain, but you have like like a lamina. You have a lamina, like a filamentous layered structure in your synaptic cleft, which has acetylcholine esterase on it. Acetylcholine esterase is a serine kinase, right? So it has this catalytic triad in it that will then cleave this ester bond, and you will have your choline and your acetyl separately. So you break it down in your synaptic cleft. Different to acetylcholamines, which are often reuptaken. You do not break them down in synaptic cleft. Acetylcholine you break down in your synaptic cleft. This is important for drug targets later down the line, especially in peripheral nervous transmission.
Okay. So you mentioned something very important, which is right here, which is the movement of acetylcholine into the vesicle while H plus protons move out of the vesicle.
Yeah.
Explain that kind of pump mechanism. What would you call it? Because it's not quite an antiport system because you're not using energy.
You are using energy.
You are. How? Why?
Your hydrogen ions are being actively transported into your vesicle. Endosomes and vesicles are often quite acidic for this reason. You're pumping them into your vesicle. So what this creates, you've got a gradient here. Your hydrogen ions want to move out. So how can you use this? You can use an antiporter, co-transporter to move your hydrogens out and bring your acetylcholine in. That's how you can set up your vesicle.
Okay. So,
if you want to know more about drug targets, you can use Vesamicol to block the VHCHT.
Okay, so what is the vesicle?
Simply put, a vesicle is a unit. Okay? It's a unit, you have a phospholipid, it's kind of like a cell within a cell. It's not a cell. It's got a phospholipid bilayer membrane. Got a membrane, and inside it, you've got something you're storing. Different vesicles do different things. You can have lysozymes, they are basically a vesicle. They've got hydrolytic enzymes in them. They're an organelle. Vesicles present in so many forms. They basically allow for the movement of a substance which you're containing within this space. So in this case, it would be a neurotransmitter, for example, acetylcholine, you've got it in your vesicle. You've got this bilayer membrane, which will then, with other signaling molecules on it, things like clathrin, often coats vesicles, really just where it's going to go and bind and then get released.
Okay. So a bit of the backstory into the whatever you're transporting in the vesicle. Now, whatever you're transporting in the vesicle does it have to move through the Golgi apparatus first?
No.
So where does the vesicle come from?
The vesicles, it really depends. It really depends because the vesicles you produce from your Golgi apparatus would be to exocytose some protein. Could that be the case here? Yes, it could. But remember, we have this pump that's bringing in acetylcholine for hydrogen. So you're producing your acetylcholine cytosolically, and then from the cytosol, you're taking it into your vesicles. So this vesicle could get pinched off from anywhere. Anything with a membrane, endoplasmic reticulum is a common one. Could be from the Golgi. But the thing that you need to remember is you're not putting the acetylcholine in from the vesicle within. You can very easily transcribe and form the acetylcholine in the Golgi and then put many of that in the vesicle. You're not doing that.
Mm-hmm.
You've got your vesicle already made and then you're putting your acetylcholine in. This is important for your vesicular recycling because it's more often than not that the rate limiting step of this whole cycle of moving acetylcholine in and out is getting your acetylcholine and getting into that vesicle.
And this process is a secondary active transport mechanism.
Yes.
It's
indirect.
Calcium ions stimulate specific proteins to bind. Synaptobrevin, binding to synaptotagmin, as well as synaptaxin, binding to SNAP25. Now, how does calcium do that?
I'll get up another slide for you. I don't know if you're going to be able to see this, but I think it's very easy to show this diagrammatically. I can explain this to you. So, on your vesicle, you have your V snares, your vesicular snare proteins, and on your axon terminal, you have your T snares. Snares basically stand for.
Let's get I have this one. Soluble
NSF, N-ethyl maleimide sensitive factor attachment receptors. So you have your V snares, you have your T snares. Your vesicular snares will be this synaptobrevin. They will be on around your vesicle, and then on your membrane you will have your T snares of your SNAP25 and syntaxin. Your synaptobrevin will bind to your SNAP25, and then it will dock the vesicle on the membrane, and then it will release the contents. The two membranes will fuse. Phospholipid bilayer will form two separate bilayers, will fuse, and then you can release your acetylcholine.
How does it fuse?
The membranes literally fuse. When you bring you got double phospholipid bilayer and then another double phospholipid bilayer. You bring them close enough, they'll interact, and they'll separate. So instead of having a circle and like a line, if we imagine it, you'll have like an omega shape.
Some of those interactions could be explained by tunneling.
Yes, they could. There's a lot of room for error here. Exploration. You could easily have quantum tunneling, acetylcholine molecule, neurotransmitter getting through that membrane even without need for vesicular fusion, existing in simultaneous states.
Depending on how big it is,
yeah, depending on the molecule.
Depending on the diameter. We will look into that for sure. So, acetylcholine has just been released from the cell in exocytosis. Yeah. Now, how is acetylcholine moving? Is it diffusing very rapidly across? How does it diffuse rapidly? How do we ensure that?
Physical factors, chemical factors, the kinetic energy in the system, really. How it moves, where it moves. You've got structural proteins there. Maybe they aid it towards the receptors. Your receptors are being upregulated often on the postsynaptic membrane. You've got all these scaffold proteins bringing them up to the surface. That's all I can say for the acetylcholine to bind to. Fission release. Okay, don't bullshit me with the fission release. Like, come on, what a joke is that as a reason for why it moves quickly? Small molecule size. Okay, high concentration gradient. Okay. Yeah, much higher than, like, it's just little things. A fission removal, so it just maintains this gradient. It's just diffusion having a gradient, really.
And are multiple vesicles being released?
Yeah. So, I think there's cats that did an experiment just after showing that calcium-dependent exocytosis was needed of these vesicles to really depolarize the muscle and elicit a contraction. He also picked up mini end-plate potentials. So those are depolarizations of the muscle even when there's no stimulation. So you are always secreting acetylcholine at your neuromuscular junction, one vesicle at a time. So you're getting very small reads, very small spikes, until you get a calcium influx, and then you release a lot of vesicles, which will then cause your big spike and cause your depolarization, at once, of your postsynaptic membrane, of your muscle, elicit contraction.
So how come the vesicles are moving one at a time, and then in other scenarios they're moving all at once? When does it move all at once specifically?
All at once, you need calcium to really move it all at once. You need to sense your calcium with your syntagmins. You need your vesicle to move along your cytoskeleton to your membrane. You can release vesicles one at a time. There's always this baseline. The way that it works, the whole human body, you always have a baseline. Graded potentials. You have your resting membrane potentials, you're bringing them up a little bit at a time, you're summating it. Your heart rate, you always have a sympathetic tone. Baseline. It's all on setting a baseline, and then it's all about controlling that level. Controlling that homeostasis.
Do you know the diameter of the vesicle?
Not to my knowledge.
40 nanometers.
Any acetylcholine that binds or even doesn't bind will be broken down by acetylcholinesterase. That's a given.
In the synapse.
In the synapse.
Okay. And if it does bind?
You'll have your receptor conformational change. You'll have your cation influx. Still need to break it down. Acetylcholinesterase inhibitors, very potent drugs. You can have all these organophosphates, horrible poisons. Or you're overstimulating both your sympathetic and your parasympathetic nervous system and your neuromuscular junction. You have short-lasting, medium and long-lasting acetylcholinesterase inhibitors. Potent drugs if you have deficiencies in acetylcholinesterase. Donezepil, used clinically for Alzheimer's. Alzheimer's is characterized by a decrease in acetylcholine. You're trying to prolong its life in that synapse. So that's really what all there is to it. Acetylcholinesterase will break down the acetylcholine. You don't want to have too much acetylcholine. Acetylcholine acts very quickly. That's the good thing. You want to have quick, transient, sharp, powerful responses for contractions, for parasympathetics. You don't want to have this sustained. Having something that's sustained, you cannot control, you will not be going back to your baseline. A lot of clinical implications for that.
4-alpha helix protein complex that consists of the 3 proteins, synaptobrevin contributing 1 alpha helix, syntaxin contributing 1 alpha helix, SNAP25 contributing 2 alpha helices. It's quite interesting. And these alpha helices is what brings about the structure of the complex. Now, did you know that the misfolding of this protein complex is known to cause neurological conditions such as Huntington's disease?
That's very interesting. Huntington's is characterized by a trinucleotide repeat, and it does cause this progressive degeneration and a lot of motor symptoms. So yeah, that's really interesting. Is this a susceptibility thing where they found these mutations leading to Parkinson's, or is this a given? Is this within the gene mutation itself, or is this a subset of the population?
I think it's within the gene itself, yeah. This is a gene mutation.
Because what happens in Huntington's is really amplify up this repeat sequence within the gene.
Mm-hmm.
So.
And are there any ways to treat Huntington's that we know of?
No. You can try and control the movement. Not really. I wouldn't know. It's kind of an inevitable. With a lot of these new degenerative conditions, you can only really try to some extent to slow down the rate of degeneration.
So a key point of innovation is finding cures to neurological disorders. This would be a huge step.
Yeah, well, how are you going to do that? How are you going to stop this inevitable process? The only way to really get to the bottom of it is through the genes. Gene editing, CRISPR-Cas9. These genomic technologies are really the only thing where you can change someone's individual genome, their DNA, and everything that's driving these processes. But it's a long road. It's a long road. There's a lot of innovations on the way. There's been a lot of innovations already. It's a long road.
Some drug treatments can definitely have an impact. Specifically, the protein subunits could change them in a specific way, which could help. That's something we may need to look into.
I don't think there are any current ways of targeting these complexes at the moment. The thing with a lot of drugs is you need to have a distinct target, and finding that target can be hard. A lot of drugs, a lot of targets, but also things like delivery, things like that. How are you going to get it there? How are you going to make sure it binds to these proteins? How are you going to ensure that they fuse and you're able to secrete your coat?
Mm-hmm. Well, certain molecules can cause conformational changes on the protein subunit called allosteric modulators. We've reached the postsynaptic membrane. How are we going to activate ligand-gated channels, voltage-gated channels? How are we going to activate these channels? What's the first step?
What membrane are we talking about?
Postsynaptic.
For what? Are we talking to the neuromuscular junction?
Yeah,
are we talking in autonomic ganglia?
We'll go for the neuromuscular junction.
Okay. In a neuromuscular junction, your acetylcholine has been released. It binds to your nicotinic acetylcholine receptors, which are ligand gated. You bind two molecules. Your nicotinic acetylcholine receptor is a pentamer. So in your muscle, the specific conformation will be two alpha 1 units, one beta 1 unit, and then a delta E unit. Two delta 1. Let me just double check that. Pretty confident, but you can never be too sure, and you never really want to spread misinformation.
Are we talking about the ligand-gated channel?
Yeah, nicotinic acetylcholine.
They are pentametric, meaning they are composed of 5 protein subunits: an alpha, a beta, a gamma, and a delta subunit.
It really varies. They are pentamers. Their conformation changes. So, in the brain, you have 2 alpha, 3 beta. And now I'm just really trying to check what it is specifically.
And so acetylcholine here will bind to a specific type of receptor on these ion channels known as nicotinic receptors. It means they're specific to acetylcholine. And it will allow ligand-gated channels.
Yeah, alpha 1, 2, beta 1, delta E. In adults, you've got, in neonates, you have the alpha 1 twice, beta 1, delta gamma, and 2 molecules of acetylcholine bind to. Okay, so all of that for my original answer. So we can cut that out.
So we've just activated the ligand-gated channel.
Yeah. So then you get your cation influx: sodium, calcium, a little bit, potassium. This is really because your nicotinic acetylcholine receptor, a lot of ion channels have a selectivity filter. Now, for cation channels, especially here for ligand-gated channels, the selectivity filter is a little bit weaker, so it will allow other ions in. It's not so selective to a specific ion in particular. So you get your positive charged ions in, you depolarize your membrane, and you'll reach your threshold value. Once you reach this threshold value, you will open voltage-gated sodium channels. The sodium will come rushing in, and you will depolarize your muscle. That's the depolarization of the muscle. That's all it does. The repolarization is then another story, and the coupling that's here, excitation, contraction, coupling, is again another story. You have things like ryanodine receptors, you have chloride ion channel, you've got chloride currents, you've got calcium sensors, ryanodine receptors, that all will function. Well, the chlorine will repolarize the membrane for another reason. You've got your T-tubules, blah, blah, blah. You've got your ryanodine receptors, which are basically a calcium sensor. You will open them, and you will secrete calcium from within the sarcoplasmic reticulum. And as you know, calcium will then go and bind to your troponin C. It will then dislodge the tropomyosin, and you'll reveal your actin binding sites, your myosin binding sites on actin, for then initiate your contractions.
Okay, so essentially we've had the influx of sodium, and that stimulated voltage-gated channels, yes, which causes the big depolarization wave. Now, do you know the specific gating mechanism of the voltage channel? The first step of that is, what is the voltage channel composed of in terms of its protein subunits?
What, the sodium channel?
Yeah.
That's last year's content, man. That's long gone out of my brain. But basically, you've got hydrophilic residues within your sodium channel. You create this kind of barrel structure, your hydrophilic residues, and when your sodium comes into this, it's small enough. You have bigger ions like potassium, they'll come in. They won't be big enough to interact with these hydrophilic residues, but your sodium It's small enough so it will interact with this hydrophilic residue and it will lose its water. I should mention that the sodium is an ion. Water, it's polar. It will form around this. So your sodium is encased in water. So you've got to strip your waters off. So when sodium interacts with hydrophilic residues, strips the water off, sodium moves through. Bigger ion potassium comes in, it's too big. It cannot interact with these hydrophilic residues in this barrel, it cannot get through. And with this water around it, it's too big to actually come through the channel. So then it cannot get in. So that's how you get your sodium moving in.
Okay, so this alpha subunit, so this barrel is the kind of channel, the subunit of the matrix.
Do you know the specific subunit?
Yeah, so it has a modular architecture. It has 4 membrane-embedded domains, each with 6 alpha helical segments. The S4 segment acting as the voltage sensor, specifically.
Do you remember? Yeah.
Now that's very important, the S4 segment. But furthermore, the domains form a specific structure to create a central pole whose structure continues to determine the selectivity and conductive properties of activation gate. Selective to sodium. Okay, the beta subunit consists of auxiliary proteins that modulate kinetics and pharmacology.
Anti-epileptic medication will bind similarly. So, another important thing to point out about the sodium channel is you do have this this kind of ball and chain on it, where you have this intracellular domain that once you've depolarized and your sodium channel has really opened and all your sodium has moved in and your membrane potential is now very, very positive, probably like plus 40 millivolts or whatever. This ball arrangement, this globular protein domain will kind of block the channel. Your sodium channel gets inactivated. This is important for something called the refractory period.
So the S4 segment is rich in positively charged amino acids. Yeah. Now, we'll get into the why this point. So that when the membrane is depolarized and becomes less negative, the attraction forces between this segment and the membrane weakens. At sufficiently negative or heightened voltage levels, basically, when the charge starts going up, it weakens this attraction force. So there's like a hinge-like action between the S4 and S5 linker, which allows the activation gate to open up. So you could target specifically the S4 subunit.
Well, if you did target it, you would basically block its voltage sensing capabilities. So if you reach threshold, you wouldn't fire. Simple as that.
Very important.
That could be quite useful. In epilepsy, you've got a lot of sodium depolarization, a lot of neuronal firing. That would decrease the rate of your firing.
So we have activation and then inactivation. Is there a step known as deactivation, which will reset this mechanism?
Yeah. Once your voltage would then reset back to your resting membrane potential, once it goes back and you don't have this, so it's really the very positive charge, the very depolarized cell state that causes this conformational change and the blocking of your voltage gated sodium channel. Once you're able to reset your voltage to more negative values, then it disassociates. You'd be probably breaking your hydrophilic, hydrophobic, your ionic interactions. All of this stuff is dictated by the membrane voltage, and your ions around will dictate whether you have this binding in this inactivation state, these forces holding it together, and then you'll be able to release this ball, as it's called, and your sodium channel will then be in its closed state. So your open and your closed state. And then you'll be able, if you have enough of a stimulus, to depolarize the cell again, open this channel if you reach threshold.
Okay, now a little shift, a little curveball here. What would be the action of the mechanically gated channel?
So what we're doing here is, you're really looking at a membrane. These channels will allow ions to pass through this membrane if you put stress on the membrane itself. You put pressure, you put tension, you are stressing these molecules. So if you have this channel and you put pressure on the membrane dynamics and interactions of that channel with that membrane will allow it to open, and you will get, let's say, sodium influx, you'll get a depolarization. That's what happens with your Pacinian corpuscles, for example, in your skin. It's where you can feel pressure, you feel vibration. You press, you're able to open these channels from the mechanical force that will move, change the membrane dynamics, change the receptor dynamics, change the conformation, change the receptors, and then allow ions to move in. So that's your mechanically gated ion channels.
Okay. So now have we got to the membrane being completely depolarized?
At the neuromuscular junction,
Yeah.
if I remember so correctly, yes.
So the next step would be exploring how the muscle actually contracts. Should we get into that, or should we leave it at synaptic transmission?
Alright, so yeah, I think we're going to leave it at synaptic transmission for this episode. Thank you for tuning in. Yeah, check out our other videos. We provide a plethora of content: maths, physics, biology, chemistry, medicine, pharmacology, but anything you've heard of, we do it. Alright? So this was our first episode of
The nervous breakdown.
The nervous breakdown. Alright? Nervous breakdown. Alright? Nervous breakdown, man. Okay, take it.
Thank you, Nicholas.
Thank you for
Tuning in.
organising this. Please make sure to like and subscribe, and catch us in the next one.